Hey learners, this is Shajata Fatima from the Department of Microbiology here with you on a new topic of microscopy. In this video, we are going to learn about the introduction to microscopy, optical microscope, classification and types of microscope, parts of microscope and general applications of microscope. What is a microscope? A microscope is an instrument which is used for the observation of small materials or small microorganisms which are not visible for naked eyes. The study of microscope is called as microscopy. Microscope works on a principle of magnification where the image of the object is magnified so that it can be seen. The substance which you observe under the microscope is called as a microscopic substance. So the microscope has wide variety of applications in the field of microbiology, pathology and different life sciences. So there are basically three types of microscope: optical microscope, electron microscope, and it is a new type of microscope which is the X-ray microscope. What is an optical microscope? Optical microscope is a basic microscope which began with the discovery of Antony van Leeuwenhoek. The light microscope uses the visible light as the source of illumination, whereas the light here passes through the series of glass to produce a magnified image. So the object is placed on the stage and directly the object is viewed through a couple of eyepieces in the microscope. It uses the optical power of the lens or the group of lenses for the magnification purpose. There are two other types of microscope, which is simple microscope and a compound microscope. Simple microscope is a microscope which uses a set of lenses to enlarge an object through angular magnification, which gives an enlarged image. It uses the optical power of a single lens or group of lenses for the magnification. What is a compound microscope? A compound microscope is a microscope which uses a series of lenses to achieve greater magnification. In the compound microscope, the one lens enlarges the image produced by the other lens. There are two lenses here in the compound microscope. One is the objective lens, another one is the magnifier lens. The objective lens is near the object which focuses the real image on the object, and the second lens, here comes the magnifier lens. It enlarges the image produced by the objective lens in a inverted phase. A compound microscope allows the exchanging feature of the objective lens, which is helpful in the adjusting of magnification. Simple microscope and compound microscope pictures are given here. This is simple microscope and this is compound microscope. In the simple microscope you can see one lens, whereas in the compound microscope it is two lenses present. The eyepiece, the components of the simple microscope include the eyepiece, the nose piece, the objective lenses, the specimen. This is a specimen holder which holds your slide without moving, a stage, a body where you will take up the microscope, a source of illumination which is the light present, and the base. Whereas when you come for the compound microscope you have two lenses here. One is the ocular lens, the head you have, the arm, as usual the body and the arm are the same, the aperture, stage controls, the adjustment, fine adjustment and the coarse adjustment to for the better magnification, brightness adjustment, and the source of illumination is the light. You have a light switch here, stage clip, objective lens is present here. Electron microscope. Electron microscope is a microscope which uses electron beams as a source of magnification. So the electron microscope basically gives a better way magnification when you compare to light microscope. It gives 2000 times higher magnification. The electron microscope is very useful in studying virus, bacteria and ultrasound of cell organelles. There are two types of electron microscope. One is a transmission electron microscope, it is called as TEM, and the other one is called as scanning electron microscope, which is called as SEM. So as the name indicates, a transmission electron microscope, the electron beam is transmitted into the ultra thin object. So that you should know that the transmission electron microscope, the specimen should be thin, so that the electron beam which is passed penetrates through the sample. Whereas in the scanning electron microscope, the electron beam scans the surface of the object and it conveys the image to a TV screen. So that you should know that the scanning electron microscope, the sample may be thick also, because it's just going to scan the layer of the sample, whereas in the transmission electron microscope, yes, the sample slide which you keep should be very thin so that the electrons pass through the sample. Here it is the images of transmission electron microscope and the scanning electron microscope. So X-ray microscope. X-ray microscope is a new technology where the detailed imaging of subcellular organelles like nucleus, chromosomes are seen. The X-ray microscope uses electromagnetic radiations in the X-ray band to produce a magnified image. The X-ray microscope exposes a film or uses a charged couple device, which is CCD detector, to detect this X-ray passing through the specimen. This is the image of X-ray microscope. Classification and types of microscope. This flowchart will give you an overview of the types of microscope. So the microscope is basically classified into two types, the simple microscope and the compound microscope. The simple microscope is just simple, there has no classification, whereas the compound microscope you have two types. One is the light microscope and the other one is the electron microscope. There are four types of microscope which comes under the light microscope, which includes phase contrast microscope, fluorescent microscope, dark field microscope and bright field microscope. You have three classifications under the electron microscopy, which include transmission electron microscope, scanning electron microscope and confocal electron microscope. Every these types of microscopes have specific usage depending on the nature of the specimen and the type of microscope is employed. The magnification of these microscopes depends upon the lens, which produces different images with different magnitude and with different resolution. The parts of microscope include two types. One is the structural parts and the other is the optical parts. The structural parts includes the head, base and the arm of the microscope, whereas the optical parts are the parts which is used to view, magnify and produce the image. They include the ocular lens, eyepiece, objective lens, nose piece, adjustable knobs, stage, aperture, microscopic illuminator, condenser, the diaphragm, condenser focus knob, Abe condenser, the rack stop, stage clip and the light switches. So this is the parts of microscope. You have ocular lens in the case of one single lens in the case of simple microscope and two lens in the case of compound microscope. So the ocular lens is a cylindrical which contains two or more lenses, and these lenses are held at the correct space. These lens helps to bring the object into focus. Next comes the revolving nose piece. The revolving nose piece holds the objective lens in a place and they help in the rotation of lenses. You can change the lens easily with the help of the revolving nose piece. Here comes the objective lens. The objective lens plays a main role in the magnifying the object. There are three objective lens present, which is 4x, 10x and 40x. Now it's like 100x is also present. The stage holds the specimen here, so the microscopic slide is placed in the stage and the stage contains two clips which prevents the slide from moving around. Next comes the diaphragm and the condenser. The diaphragm and the condenser control the amount of light which passes through the slide, so it also plays a dual role in the magnification of the image. The light source or the mirror. In light source, it provides a source so that the object is viewed. That's all. The frame is like stability to the microscope. It is used to hold the microscope. It is U in shape. The fine adjustment screw. The screw is helped in the final and clear focus of the object, whereas the coarse adjustment screw helps for the initial focus of the object. The structural parts include head. As I told, the head is the body of the microscope. It carries the optical parts of the microscope. When you see the base, it supports the microscope and carries microscopic illuminators like mirror, source of illumination, no? Mirror, light, etc. Arms are just the connecting base of the head and the eyepiece tube. It supports optical parts of microscope include eyepiece, which is ocular, which is used to view the specimen. The top of the specimen standard optimization is about 10x. Optical microscope has 5x and 30x. Eyepiece tube. Eyepiece tube is the eyepiece holder above the objective lens. In a binoculars, the eyepiece tube is flexible and rotatable for maximum visualization. This is mononocular, they are not flexible. Objective lenses are major lenses used in the specimen visualization. The magnification of the objective lens is 40x to 100x. 1 to 4 objective lens is present in the microscope. The nose piece. Nose piece is a revolving turret which holds the objective lens and it is movable. The fine adjustment knobs and the coarse adjustment knobs are the types of adjustment knobs in the microscope which is used for the initial and final resolution and magnification of the specimen. Stage is the place where the specimen is placed and it has two clips which prevents the slide from moving. The aperture is just the microscopic stage hole which transmits the light source. Microscopic illuminator is nothing but the source of the microscope, light source. Condenser is the lens that collects and focuses the light for the illuminator specimen. So the diaphragm controls the amount of light which reaches the specimen. Condenser moves up and down so that it controls the focus of the specimen. The Abe condenser is designed for the high quality condenser which is movable. It allows high magnification about 400x. The rackstop controls the stages and prevents the objective lens from getting close to the specimen. The applications of the microscope include: it helps in the examination of the specimen by getting larger view of bed sample in detection of the foreign pathogen, parasites, bacteria and etc. The studying of the microorganisms, cells, crystalline structures and molecular structures is done, helpful in analyzing and diagnosing the tissue samples. The compound microscope is cost effective and is useful in teaching schools and colleges. Blood cells, cheek cells, parasites, bacteria and other thin organs of the body, thin sections of the organs, UTI and urine samples are analyzed. It is useful in the detection of criminal cases, discovery of organisms. Thank you for your patience on my lecture. For any queries, contact shasayology024@gmail.com. Thank you.